content for next year for biology?
I'm sorry, could you please repeat that again?
Where do you want to be in terms of content next year for biology?
So like in that sense, I want to be ahead of my class and like know my content in depth, so I'm able to answer the questions more better compared to this term.
Good, but like where exactly? Like mod 6, mod 7, mod 8, where do you want to be? Because we need to have
Mod 6.
Okay, good. I would agree. I've shared my screen. I'm going to pop up Notability, and let me just explain to you what my game plan would be, and you guys can fine tune it based on your individual goals. So, 1 second. Okay. Okay. Okay, number one, let's talk about content. Right? I would expect all of you learn ahead and get to the end of mod 6. That's where you'd ideally like to be in Feb, because you'll be starting mod 6. If you're a module ahead, you can comfortably stay ahead throughout the year because things are going to start going ahead. You're going to get assessment tasks which will slow down your content pace. End of mod 6 would be my checkpoint in terms of knowing the content. Okay? We'll be doing it in a very fast-paced manner. If all of you can get here by end of Jan, I can get through mod 6 in literally 4 or 5 weeks. It is a short module and it is a supplement to mod 5, genetic change. Right? So that would be my content checkpoint for all of you. How will you do that? I'm going to give you guys all access to the mod 6 booklet. Now, can you guys raise your hand? Who's been writing their own module notes on top of the theory booklets I've been giving you? Okay, that's good. Everyone learns differently. Some people like to always write their own notes. I would say go for it. In that case, you need to get that booklet done. Those of you who aren't writing your own module notes, you have the benefit of time at the cost of potentially having tiny gaps in your knowledge just because you're reading the notes versus creating it yourself, right? So in that case, in terms of past papers, do you guys remember that Mod 5 Bio additional questions booklet that I've given you access to? Additional questions. I would want you to complete that entire booklet. Okay? So the correlated trial questions for Module 5 that I've organized for you, complete that, and also complete the edX booklet questions. Good. Now, who has a timetable? Can you guys hit the raise hand icon? Now, I know I haven't talked about biology content just yet, and it's because this stuff is arguably more important because it's intentional planning than just jumping straight and learning as much content as possible, right? So, can you guys hit the raise hand icon? Who has a timetable or a routine that they've set out for the holidays? Okay, cool. What I'm going to do, I'm going to help you all out. I've created a holiday habits timetable calendar for myself, and I will share this access to you guys. If you don't use Notion, I would highly recommend it. It's probably one of the best organizational apps you can use. So literally, I'll send you all this template. Feel free to use it. It's basically a calendar. Every day, it has a checkbox. I've written my checkbox of what I like to do. You can see on the tent. And then what it does is it gives you a little bit of a progress bar on how consistently you've been doing that over the holidays. So set aside 5 or so habits for yourself. I'll create a template for you based on your habits, and I'll send it all to you guys probably by the end of today. But make sure you have a bare minimum routine that you're following. The last thing I would say is broader in terms of HSC. You want to be doing the same subject about every 3 to 4 days. Okay? Which ends up being about 2 times a week that you study a particular subject. That's what your study block timetable will be. Okay? Good. Does anyone have any questions? Because I can't stress how important this holiday break period will be. Have fun, enjoy, but after New Year's, you will have to start doing a ton of practice questions, fine-tuning, refining, and learning the knowledge. Good. All right. Good stuff. I remind me to send you all the timetable as well, and we'll get onto that. Okay, so I guess in terms of content, what did we learn 2 lessons ago? Let's go to Alyssa. What did we cover?
I think we went through DNA replication.
Yeah, very good. Okay. Now, talking about DNA replication, you've all learnt about the various phases. I'm sure we've spoken about mitosis, meiosis in sufficient detail. Let me just quickly quiz you guys and see how much we understand. I know, Kriti, you finished Module 5 as well. Kriti, when we're talking about meiosis, what are the 3 parts of meiosis that lead to genetic variation compared to mitosis? What are the 3 unique events that occur? Any idea, Kriti?
Crossing over, independent assortment, and random segregation.
Good, right? And this is a question that I like to hone and ask again and again because this is the bane of students' knowledge difficulty. What is the difference, Kriti, between random segregation and independent assortment?
Isn't it like one with the pairs, like the chromosomes, and then independent assortment's like with the chromatids?
Very close. Your first point is completely right. I'm going to pass it to Anne. Anne, can you just quickly remind the class, what's the difference between random segregation and independent assortment? And before you do that, Anne, I'm just going to use a little diagram to help everyone out so we're not all confused about this. Okay. Anne, take it away.
Was it for independent assortment? It's like the segregation of the homologous pair, and like it doesn't affect the other homologous pair. So the separation of one homologous pair doesn't affect the order, or like the way the other homologous pair orientates itself.
Very good, right?
Whereas
So
for random,
yeah, keep going.
for random segregation, it's like the random movement of the chromosomes, like within the homologous pair.
Very good. So random segregation within a homologous pair is the most important thing you can all remember. Within a homologous pair, chromosomes will always move to opposite ends. They can move in this fashion, or the left chromosome can go here, which means the right chromosome has to segregate in the opposite direction. That is random segregation within a homologous pair. Independent assortment, just like Anne mentioned, is that the random segregation of the homologous pair circled in black is independent or completely unrelated to the random segregation of the second homologous pair underneath. That is it. Okay, very good. And finally, let's go to Ruthania. Ruthania, can you remind the class, what is the effect of random segregation? What is the effect of independent assortment in terms of alleles and genes that an organism inherits?
It leads to genetic variation. So the
It's a hard
swapping of
question. Yep, keep going.
the swapping of the alleles inherit different genes in order for the.
Okay, so we need to be very specific here. Which one does which? So, Alyssa, I'm going to pass it on to you. Alyssa, when we talk about random segregation, we're talking about one homologous pair. Do homologous pairs have different genes or the same genes?
They have the same genes but different alleles.
Very good. So, of course, the effect is if these chromosomes within a homologous pair that have the same genes but different alleles move to different directions, that means that alleles that can be inherited by a gamete, the allele combinations increase. Because whichever direction that a chromosome goes, that will eventually go into form one gamete. Whichever direction that chromosome goes will eventually go on to form another gamete. So this increases the allele combinations a gamete inherits. Full stop. Good. What about independent assortment? Alyssa, I'll let you take it away.
In independent assortment, the chromosomes of different homologous pairs, they separate independent to one another. So one will go to one side and the other one will go to the opposite side.
Very good, right? And so the effect of that, different homologous pairs, Alyssa, did they have the same genes or different genes?
The different genes?
Different genes, exactly. So this homologous pair would have, let's say, the gene for height this homologous pair would have the gene for hair color. As a result of that, right, the inheritance of one gene is completely independent to the inheritance of another gene. AKA, I can be short and have brown hair. I can be tall and have brown hair. They don't have to be linked. Okay? Very good. Good. Guys, I'm repeating this again and again. It's because there are very specific parts of this module that students will always find challenging, and I don't want you guys to do that for this coming year. The other stuff, DNA replication, very straightforward. If you remember the enzymes, you're quite set. And all the extension stuff I taught you, it rarely ever comes up in exams. But like I told you, meiosis is probably one of the highest yield topics, not just in Module 5, in the entirety of biology. I promise you in your exam, you'll get a model about meiosis, asking for the issue, and then an improvement. It's usually always in an MCQ or in a long response. Very good. So we've gone through DNA application. I'm not going to go through that again. What we're going to talk about is something far more interesting, which is the story of how your DNA, that instruction manual, codes for this, the living organism. How does the letters A, T, G, and C allow me to have tissue, muscle, ligaments, an entire phenotype, a physical appearance, characteristics, disease, non-disease, etc. Right? Essentially what medicine is it's all about treating the phenotype, the physical characteristics. And that is coded for entirely by your DNA. So that leads us to inquiry question 3. Now, we've already briefly spoken about it. When you get an inquiry question, you want to answer it. We've already answered why protein synthesis is important. It's because your entire body, right, consists of chemical reactions occurring in an organized manner. And I'm sure you all learned in year 11, Kriti, what do we call biochemical catalysts that speed up the rate of cellular reactions? What are their names? What's the name of that description or definition? Biochemical catalysts that speed up cellular reactions. Kriti? Any ideas?
I can't remember.
That's all right. Enzymes, right? So I want all of you in red. If that's not the first thing that comes to your mind when you think of protein, it should be, right? When you talk about why polypeptide synthesis is important, it's because when we make proteins from DNA, the vast majority of them will form enzymes, right? Life would not occur if we did not have enzymes. For example, I'm sure you all know, we eat glucose, we break it down to make ATP energy, and that's like the energy currency of the body. It is how the body allows chemical reactions to occur, things to be built up, waste to be taken away. We use about 100 plus different enzymes to allow those reactions to occur. Okay? Without those enzymes, the reactions would occur too slow. We wouldn't make energy at a fast enough rate to sustain life. You have death. In fact, when an organism dies, we can always link it down to the point where enzymes stop working. Very good. Now, if a protein is not acting as an enzyme, okay, it doesn't have a functional role, it's acting as a structural role, right? So in the case of bone, does anyone know what bone is made out of? This stuff is hard, right? If I hit bone on the table, very hard. What is it made out of, Matthew?
Calcium.
Good. Calcium makes up about, I would say, 40% of bone, right? Bone is living. It has cells. They're called osteoblasts, osteoclasts, osteocytes. There's also protein, everyone. There's collagen protein. The same protein that makes up your skin, those tissues, is also present in bone, and it gives it some degree of flexibility. Every structural part of your body has some element of protein. Collagen in your skin, collagen in your bones. I think I've already written collagen. I'll write it here. Collagen, right? In skin, bone, and other connective tissues. So connective tissues meaning ligaments and tendons, right? Who can think of some other structural or functional roles of proteins? What's an enzyme that you all should use as an example when we talk about enzymes and proteins? And what enzyme are you going to discuss?
Isn't there an enzyme called like lactase or something?
Yeah, there is lactase. So if you drink milk, lactase in your digestive system breaks down the sugar lactose. If you don't have the enzyme, you don't have the gene to make the enzyme lactase, you're not going to be able to break down lactose. And if you understand basic osmosis, you'll understand why people get diarrhea if they're lactose intolerant. The lactose sits here in the digestive system, it draws water in, and then you end up with diarrhea, right? So that's lactose intolerance in a nutshell. Okay, so that's one. Let's talk about some more important ones, because I think lactose you could easily supplement with pills, right? And a very important one is catalase, right? I'm sure you all did those enzyme reaction rate experiments in year 11, but I'm not sure if you grasped how important catalase is. If you don't have catalase, all of us will end up getting cancer and dying. The reason being, catalase breaks down a very harmful byproduct from chemical reactions in the body known as hydrogen peroxide. We call it peroxide, right? And it will break it down into the harmless products, water and oxygen. That is what catalase does, right? This compound here will rip electrons off molecules like DNA, causing mutations leading to cancer and death. So this is arguably one of the most important enzymes in the human body you studied last year. Okay? Very good. Okay, guys, I want us to brainstorm. What are some other proteins? There's way more. There's more than you and I can brainstorm in a year in terms of proteins in our body. So, Matthew, any other proteins you know that are important?
No, not really. I don't remember any.
That's all right. And
Are antibodies one?
very good. Good stuff. Is anyone doing module 7 right now in biology? The order you do biology does not matter. It's just if you do 5 and 6 together, 7 and 8 together. Good. Brittany, what are antibodies? I think antibodies are some of the coolest proteins in existence. They're the reason you and I are alive. They're the reason we survived the pandemic. If you had COVID, they're the reason you are still here today. Brittany, what are antibodies?
Antibodies are proteins that that immobilize antigens.
Yeah, so all of you right now should be writing proteins, arrow, functional, and then structural. Functional and structural. And every example that we've discussed, you should be putting it under each category. Antibodies are example of functional proteins, right? So enzymes were another example of functional proteins, antibodies. Antibodies, everyone, I want you to remember this, because if you remember this analogy, you will do well in Mod 7. These are like handcuffs. If you handcuff a prisoner, Edward, does a prisoner die? Very simple question.
No.
But yeah, no, right? It simply immobilizes, incapacitates, and locks up the prisoner. That is exactly what antibodies do. They do not destroy bugs, they do not destroy foreign organisms. What happens is a bacteria will bind to that end of the Y-shaped molecule and get locked up by the antibody. It is a protein, okay? It is a protein we make to lock up any foreign bugs, disease, etc. Okay? Very good. There's another very important functional one. All of you guys have released millions and billions of these molecules since the beginning of class just now. They're being released in your brain. You are using it to strengthen connections, reinforce memory. What am I talking about? And
I'm not sure, but is it hormones?
close, but you're completely right. Hormones are another very good example of functional proteins. Very good. And what are the two most important hormones, and you need to remember for this module? Two most important hormones.
Not too sure.
Estrogen and progesterone.
Progesterone? Okay.
Good. All of you should be able to tell me where they're released from and what they do. I'm not going to talk about that now. We've spoken about that, and I'm sure you'll have access to the recording too. Then I'm talking about something else. Released in your brain, they're called neurotransmitters, everyone. Neurotransmitters. in essence, your consciousness, who you are, your identity, can be broken down, theoretically, into the firing and non-firing of brain cells called neurons. What they literally do is they sit and they connect to each other like this. And when you think of an idea, the neurotransmitter that holds that memory or concept is going to release neurotransmitter, excite this neuron, that will release neurotransmitter, excite a neuron. And that is why when you study, what all of you are trying to do is keep firing the same neurons that are associated with HSC biology. What ends up happening very beautifully is you form more connections in your brain. It strengthens, and the arms get stronger. They release more neurotransmitter until this stuff becomes muscle memory, essentially, which is how it is for me at this point, right? There's no need to study HSC biology for me. After so many years, it just becomes like speaking English almost, right? And I'm sure for all of you, that's how year 7, year 6, year 3 content that you learned is at this point, right? It's essentially what learning is. Very good. So now you should all have at least 3 examples of functional and structural proteins. So in a 6 mark, 7 marker, feel free to use any of those examples we discussed. Does anyone have any questions about the importance of proteins? Hopefully I have stressed it. Very good. Okay, Rithanya, remind the class, you've done this before as well, so how is DNA structured in a eukaryote versus a prokaryote? Rithanya?
In a prokaryote, it's like one whole loop, so there's no and it's in the cytoplasm.
Good. And what about eukaryotes?
It's like coiled up and it's in a membrane-bound nucleus.
Good. And I asked you this question a couple of weeks ago. How many chromosomes does a bacteria have?
1.
Very good. Right. So you're learning. Very good. So all prokaryotes have 1 chromosome as a loop in their cytoplasm. So it looks like this. This is literally copy pasted from a sample answer. If you are ever asked how DNA exists in a eukaryote and prokaryote, this is exactly what you're going to put. This is a chromosome. Why does it look like this? Why does it not look like this? Or like this? Anyone? Why do we not see that shape?
Is it because it goes into like the tightly coiled like form of a chromosome when DNA cell replication occurs?
Very good, right? Because I think you can all appreciate what is a chromosome. A chromosome is just histone proteins, right? These ball of proteins with DNA wrapped around it. And so when we need to read DNA, when your cell needs to read the A, T, G, and C to start making protein, the DNA needs to be loosely wrapped around those proteins. Otherwise, the enzymes can't get inside those nooks and crannies and read the letters of DNA to make the protein. So 90% of the time, your DNA actually looks like spaghetti, just like this, right? That's because the DNA is loosely wrapped around the histones, and that's how enzymes can access it to make proteins. And that leads us to the most important part of this dot point, which is modeling and understanding protein synthesis. Can you guys hit the raise hand icon? Who's already learned this, so I know how fast to go in terms of this pace? Protein synthesis. Good. Good. Okay, that's all right. So about half-half. So those of you who have already learned it, can you just keep the raise hand icon up, just so I know who to ask questions a little bit more, so all of you are learning at the same pace. Very good. All right. All right, so Ed, we'll start with you. When we talk about protein synthesis, there are 2 stages, and you see it right here: transcription and translation. Ed, what are they, and why are they called transcription? Why are they called translation? Why did we use those words?
Well, transcription is like when the DNA is copied or transcribed into an mRNA strand to leave the nucleus. And translation is turning a codon into an amino acid to form a polypeptide chain.
Okay, good. Okay, there's a bunch of big words, and I'm sure for those of you who've never learned this before, you're probably thinking, what is a codon? Which is completely fine, right? Okay, this is our little schematic. This is a cell. The nucleus is our little circle. The cell is the big circle. Everyone can appreciate that. DNA is sitting in the nucleus at this point. Now, let's take a step away from biology and ask ourselves, what does transcribe mean? Pretty, if I said, I want you to transcribe the Pearson biology textbook for me, what does that mean? Pretty?
Put into like a written form.
Copy is transcribed, right? When you transcribe something, you're copying. When you're transcribing audio, you're converting it to text. The reason we need to transcribe DNA, let's think about this. We're talking about protein synthesis here. Alyssa, what organelle is involved in protein synthesis? What did you learn in year 11?
Ribosome.
The ribosome. The ribosome is sitting all the way out here in the cytoplasm. This is a ribosome. Kind of looks like a burger. It's got 2 units. Kind of looks like a burger. So 2 ribosomal subunits. And somehow the genetic material that is the DNA needs to reach this ribosome. Now, I want you to also understand this. The cytoplasm is a very, very dangerous area. It's kind of like the US, right? A lot of enzymes that are ready to stab, destroy, shoot, destroy whatever molecule exists in the cytoplasm. It is a soup full of destructive enzymes. So if your DNA left your nucleus and went straight to the ribosome to make protein, your DNA would be destroyed. You would not have DNA in the cell would die. So then the body is very smart, right? Evolution, etc. Thought of a very interesting thing to do. And what it did was it decided, well, we don't want DNA to be destroyed, so let's make a low-quality copy of DNA that is very resistant to destruction and send it to the ribosome. And so then, who cares if that copy gets destroyed? Because the original version, right, the OG, the DNA, stays the same, has the same integrity, the same letters, no mutation, etc. Right? And so the first step in making a protein is to get a copy of DNA made. And so scientists call this process transcription because you're making a copy of something, right? So transcription is the first term. It is making a copy of DNA. And the copy goes by this name mRNA, which we're going to talk about shortly what this means. The M stands for messenger. The reason it is called messenger is because the DNA is sending a message to the ribosome. Can you all appreciate that? Hence the term messenger RNA. We'll talk about what RNA is shortly, but just remember now, it is a low quality, resistant to destruction version of DNA. Very good. Now, Ed mentioned this term translation. And what is translation? I think you put your hand up when you learned protein synthesis. So.
Yeah, so translation's when you like change something from one form to another. So like if you're translating a language, you'll change it from like English to like I don't know, like French or something.
Very good.
Changing the bases, like the codons, into amino acids.
Very good. And just for everyone, what a codon is just a set of 3 bases, right? Codon. The reason we call it a codon is because it codes for exactly 1 amino acid of the protein, or the polypeptide chain, right? Codon. That's just what codon means. We'll talk about that shortly. I don't want to introduce any new terms just yet. But the key point here is exactly what Anne said. Translate is one language to another. So what we're doing is we're going from the 4-lettered language that is DNA, RNA, and we're going to another entirely different language, which is the language of amino acids. And all this translation, it occurs at the ribosome. The ribosome is the organelle that converts the letters of mRNA into the amino acids that make up a protein. Okay? How many amino acids do we have? And where did they come from? So, Alyssa, any idea how many amino acids exist?
I think 20, like you have 21 or 23.
Good. 20, exactly 20 amino acids. Very close, but good. Okay, so we have 20. This is like a 20-lettered alphabet, right? There's leucine, histine, arginine, proline, threonine. There's a 20-lettered alphabet, and there's a 4-lettered alphabet, which is DNA. So you can see clearly they're different languages. That's the first thing to appreciate. The next thing, right, I want you all to understand and appreciate. What did we say? What is a codon again, Matthew?
It's just pretty much 3 nucleotides.
Good. 3 nucleotides that codes for 1 amino acid. Okay? Now, let's do the maths here. Okay? How many bases can sit in the first position? We're doing a bit of very basic terms and commas. How many bases can sit in the first position of a codon? Britannia? You can have an A, you can have a T, you can have a G, you can C. So, how many?
4.
4. Britannia, how many bases can sit in the second base position?
4.
4. And the third? A, T, G, and C. So, quick maths, that's 64 combinations. How many different amino acids did we say we had? 20. Now, this is a bit of a challenging question, but I want to see who's really thinking. Why is it that 64 different combinations of bases code for only 20 different amino acids? Why did the human body do this? Why is there a huge overlap? Well, the implication of this, I hope you can all appreciate, is that an AAA and an AAT, even though they're different codons or different bases, they're still going to make the same amino acid. Right? There is an overlap where multiple different codons will make the same amino acid. Why did the human body do this? You can tell me the answer, you're very critically thinking. This is the difference between memorizing, rote learning, and really understanding biology. And
isn't it to like allow for greater flexibility, so like there's less chances of proteins being made wrong?
perfect. Very good, right? So literally, I want you to imagine this. Let's say you go out to Bondi Beach, it's summer right now, you have a good time. Your DNA gets about 20,000 base mutations per hour. 20,000 letters change into unwanted ones. Your enzymes in DNA replication will fix the majority of them. So the DNA polymerases will fix 99% of them. 1% will stay. Now, let's say that change was a change from A to a T. Even though you got the mutation in the sun, which worst case would lead to, changed proteins, changed cell replication, cancer, and death, what now happens is you still make the same amino acid. And so you still make the same protein that's not diseased or dysfunctional, and you will not be affected by the mutation. In fact, this is taught directly in Module 6, everyone. This is what is called a silent mutation. I want you to remember this. The fact that multiple codons code for the same amino acid, okay, aka there is a redundancy of some codons, the benefit of that is exactly, as Anne mentioned, is for increased genetic stability and to allow for silent mutations. Good, right? All right, very good. Any questions? Okay, good. Let's go back to our story here. So hopefully now everyone understands what transcription, just as a very one-line overview, is DNA to mRNA, and everyone understands that translation occurs at the ribosome, and it is the mRNA to the polypeptide sequence. Okay. The last thing I will add is transcription. The copying of DNA occurs within the nucleus because that's where the DNA is. Okay. Very good. Now, before I tell you about the actual processes, let's talk about this molecule here, not the M, the RNA. You guys, again, who's already learned this? Just hit the raise hand icon so I know who to ask. Good. And okay, Alyssa, can you tell us what's the differences? What are the differences between RNA and DNA? And if you've learned this as well, why are they different like that?
RNA is single-stranded, and I think instead of a T, it uses uracil as a base unit.
Very good. Single-stranded. And it uses the base uracil instead of thymine. There's no such thing as a T. Now, here is a bit of a challenging question. You guys should always be doing this. When you learn something new, ask yourself why, right? So, anyone learn or ask the teacher why RNA has uracil instead of thymine? Alyssa, any ideas? Why does RNA have uracil instead of thymine? Think about what I said about RNA in the very beginning. What was the goal of RNA?
Isn't the point of it for like transcription for protein synthesis?
Yeah, the point of it is to create a copy of DNA that can survive across the cytoplasm to the ribosome without being destroyed. So uracil is just a tougher, reinforced, armored version of thymine, right? It is more resistant to degradation. That's all. That's why we have uracil instead of thymine, okay? Good. The second thing is that it's single-stranded. The third thing that you usually aren't required to ever state in an exam is the actual reason it's called RNA. RNA stands for ribonucleic acid instead of DNA, which is deoxyribonucleic acid. So what this means, ribonucleic acid, what is the ribo referring to, Edward? What part of DNA, the base sugar or the phosphate, is the ribo referring to? All right. It's the ribose instead of deoxyribose. Okay. So RNA has a different sugar molecule, hence why it's not called DNA. Okay. Instead of deoxyribose. Good. Those are the three main differences. And I want you to remember the most important one is this, and structurally the most important one is this. I don't care if you say the third thing or not. Okay. Good. Any questions so far? Okay. Let's talk about transcription. So, when we talk about transcription, very similar to DNA replication, there are enzymes involved that make this new strand. The one enzyme I want you all to remember, well, let's go back and think about this. Sarah, what was the enzyme that made new bases, or that arranged the new bases in DNA replication? What was the enzyme that did that, Sarah?
In DNA replication?
Yeah.
I'm sorry, I don't remember.
That's all right. Yeah, go back and revise that. Atul, what is the enzyme in DNA replication that makes and creates the new DNA strand?
DNA polymerase.
Very good. Now, very similar to DNA polymerase. What does polymerase mean? Polymerase means make, new. The enzyme involved in making the RNA is called RNA polymerase. I need you to state the enzyme, everyone. Enzyme involved is RNA polymerase. Okay? Very good. All right. Now, you'll often see these words sense, anti, sense, strand. Forget about it for now, because I don't care if you state in your answer or not. The key thing I need you to state is the word complementary. And I need you all to really, really understand this. When we are making RNA, we are making the RNA complementary to the DNA strand. That is always the case. Whenever you make new DNA or whenever you make new RNA, you pick a strand and you always make complementary bases. Instead of an A, you'd make a T, but since it's RNA, it'd be a U. Instead of a T, you'll make an A. Okay? It is never, ever the case that an enzyme will just copy a strand as is. Okay? It is always complementary synthesis of a strand. And that is why I stressed very early on, the whole reason why DNA can replicate is because bases are complementary. If an A coded for a T and a G, enzymes would have no clue which complementary base to make, right? So I need you all to state that in your answer. If you don't use the word complementary, you'll lose a mark. Okay? So RNA polymerase creates complementary RNA nucleotides to the template DNA strand. Word for word, that is the definition of transcription. Okay? Now I'm going to give you some additional info. This is not as important. I don't care if you remember this or not, but it does help, and it might help you for some MCQ questions. There's these terms called antisense and sense strand. The way I understand and remember this is the sense strand makes a lot of sense. AKA, this is the strand that you want the RNA to look like, because this is a strand that has the information to make the protein. The complementary strand in the DNA to the sense strand is called the antisense strand. Antisense does not make sense. Okay? So what RNA polymerase does is it binds always to the antisense strand and it makes complementary nucleotides to it. What do you all notice now? If you make complementary nucleotides to the antisense strand, what have you done in relation to the sense strand? Matthew, if I make complementary bases to the antisense strand, what does the RNA now look like? Does it look like the antisense or sense strand?
Sense.
Exactly. It looks just like the sense strand, except instead of a T, there is a U, right? And so that's what antisense and sense mean. But again, I've never seen an exam question ask about that. But like I said, must use this word. You must use this word. Good. Any questions about transcription at all? And question?
So I just wanted to make sure. So when DNA replication occurs, DNA polymerase makes the bases.
Yeah.
And then for when mRNA is made, it's RNA polymerase.
That's right.
Okay.
Polymerase, yeah.
Do they
Yeah.
also, sorry, do they also have like you know, for DNA replication there's like primers and everything? And
Yeah.
the helicase and everything, does that also occur for mRNA being made?
Very good question. I'm actually not too sure. I don't think so, because whenever I've learned it, I've never spoken about or learned about primers in transcription. They could be, because like I said, this process is so infinitesimally complex, right? You can just keep going in deeper scales and learn more. So I'm not too sure. I'm quite sure there is no primers involved in this, but key thing here is it's just RNA polymerase that you need to say.
Okay, thank you.
Good question. Good. All right. Okay. So again, like I said, you really need to understand the gist of this rather than memorizing and rote learning. Remember what I said. What was the purpose of RNA? It's to create a copy of DNA that is resistant to destruction. So one modification we've already made is that instead of thymine, we have uracil. But that is not enough. The enzyme in the cytoplasm will still eat away the mRNA even before it could reach the ribosome. And so what the body then does is after the mRNA is made, it will modify the mRNA. And this is the term or the heading for this is, where is the word? I'll just write it for you. Post-transcription modification. This is in between transcription and translation. After your mRNA is made, it is further reinforced with armor to prevent destruction in the cytoplasm. Okay? Good. Now guys, two things happen here to reinforce the mRNA. Okay? You add a cap and you add a tail to the mRNA molecule. So you can see here, forget about the words intron exon, forget about this for now. We're going to talk about that later. For now, just look at the ends, right? So this is all RNA. Okay, all of from here all the way to the end. That is RNA. What we've done is we've added another molecule that is going to be sacrificed or eaten up by those Pac-Man enzymes in the cytoplasm, and it's called a 5' cap. The reason it's called 5' is because it's being added to the 5' end of the RNA. It's made of this molecule called guanosine. So you might see that in your textbook, 5' guanosine cap. But if you just remember 5' cap, you're good. Okay. The other thing that enzymes do is they add multiple adenines to the end of the RNA sequence. And so this is called poly meaning multiple, right? Polygon, multi-sided shape, poly, multiple. A poly A tail. So we add multiple, hundreds and hundreds of adenine molecules that are also going to be sacrificed. The purpose of this is when enzymes start destroying the RNA from this way and this way, the actual important coding RNA is preserved. Any questions so far? Good. All right. Very nice. Post-transcriptional modification. Only talk about this if it's a 4-marker or more on protein synthesis. With a 3-marker, 2-marker, 1-marker, only transcription translation. This is going to help you with your trials, especially with MCQ questions as well. Okay. Good. Next. Alyssa, what are introns and what are exons? Because I think you said you learned this already.
I think introns are just sections of non-coding DNA.
Good. Right. Do you know why they're present, Alyssa?
I think we learnt it, but it was more of like extension, so I don't really remember it.
Yeah, look, the actual reason is we don't really know why there's sections in the mRNA that actually don't code for anything at all, and the cell needs to get taken out. But I'll tell you why. There's like a slight reason. Okay. But anyway, coming back to this, the way I remember this, you need to have mnemonics to understand this, right? I remember exons have an E. E for they are going to be expressed, aka they're going to be made into protein. So there are regions, there are letters, so it could be an A, T, G, C, G, going into hundreds of bases down. There could be a section of the RNA that actually does not, or is not meant to code for any protein. And so enzymes recognize that, and they cut those sections of RNA out every single time a protein is to be made. We as scientists call those sections that kept getting cut out introns, because they don't code for anything. And the sections that are left, can you see here? Now it's just exon 1, 2, and 3. We call them exons for being expressed. Okay? Now, the question I asked Alyssa was quite unfair. It was a very hard question. But the reason that we keep exons, right, or the reason we have introns and exons, we largely don't know. But sometimes what we see happening is enzymes will remove most of the introns, but keep some introns in there. So this intron might be cut out, but this intron might be preserved. Would that change the protein that's made? Edward, if I kept one intron versus removing all introns, would that change the amino acid sequence of the protein?
I'm not sure. Maybe not.
It would, right? And the reason is introns are bases. So if an intron has an AAA and a UUU, that's, remember, every 3 bases codes for 1 amino acid. So now the protein is going to be, now, very simple case, 2 amino acids longer than it otherwise would have been if we cut that intron out. And so sometimes the same gene can make slightly different proteins, right? And that's affected by which introns that we cut out and which introns we keep in, right? So we can have a variation of proteins made from the same gene by the way the introns are cut out. Good. Now, there is an organelle that does this, that cuts out I like to think of it as the scissors, the molecular scissors of RNA. It's called a spliceosome. And the reason it's called that is because the process of cutting your RNA is known as splicing. Remember, post-transcriptional modification consists of 3 things. 5' cap. Matthew, what was another thing that RNA processing consists of? What else do we do to the RNA?
Add polytail.
Good, poly A tail. And Ruthania, what's one more thing we do different? to modify the RNA?
To get rid of the introns.
Good. I need you to use the terms now. Splicing. Okay, that's the term. Very good. So those are the three things that happen to modify RNA before it's ready to leave the nucleus. Everyone with me? Any questions so far? Good. All right. Now, for the next part, I ask all students to do this. Do this in the break, or do this after the class. Write this down, because what we're learning here, these aren't words, right? They're happening right now. Things are moving. Organelles are jumping. Things are processing inside your body this very second. This is a visual process. You are not going to understand it through words. Okay? So to truly understand this next process, translation, I want you to watch HSC HSC, not even HSC. I want you to watch, it's called Crash Course Biology. I think some of you have already seen this. Protein synthesis or polypeptide synthesis. Search that up on YouTube. Do it in your spare time, your break or after class. But I remember being a student, the first time I learned this, it was hard, right? But when I watched this YouTube video and I saw how things added onto the RNA and the new strand was made, it made so much sense in my head. Okay, so go watch that video to truly understand what translation is. Okay, but I'm still going to do my best to explain it in terms of images for us now. Good. So we've already spoken about translation. Going to Kriti, what is translation again? And where does it happen?
It happens in the ribosome and the cytoplasm.
Good.
It's where the mRNA, well, tRNA with an anticodon and an amino acid gets attached to the mRNA and forms a polypeptide chain.
Good, good, right? Good. And we're going to talk about tRNA and all these new terms just now, right? Very good. So this is translation, this image here that I'm going to be showing you. First thing I want you all to do is understand it. Second thing I want you all to do is come up with an exam answer when I ask you what translation is. Okay? So let's start with understanding the process. Now what's going to happen is that mRNA is going to go and it's going to sit between the two buns or the units that is the ribosome, right? What is a ribosome made out of, Alyssa? What is the ribosome's composition?
Is it also amino acids?
It's made out of protein and RNA. There's ribosomal RNA and there's also protein. Very good. So just so you all understand, it's not actually a glob like this. If you look at what an RNA looks like on a really like zoomed in image, it just also looks like spaghetti. It looks like 2 units of spaghetti because it's RNA and there are units of protein inside of it like this. So as you can see, it looks super messy and complicated. That's what a ribosome really is, right? Okay, first thing that's going to happen is that the mRNA is going to sit between the ribosome subunits. Okay, what the ribosome will then do, everyone, is it will go codon by codon across the mRNA strand. Okay, so I want you to imagine, forget about this stuff. So let's say this is a 5' cap. Okay, let's say all of that was a 5' cap, just to keep it very. Let's say all of this was the 5' cap. So we don't actually code for it, we don't use it. Okay? So what the ribosome is going to do is going to read the first set of 1, 2, 3 bases, known as a codon. Okay? And what it will then do is it will ask for help. It will ask for help for another molecule that goes by the name of tRNA. What does the T stand for, Edward? In tRNA. What does T stand for?
RNA.
Sorry, I didn't get that. A bit louder.
Transfer RNA.
Very good. Transfer RNA. Very good. So tRNA is transfer RNA. So the reason it's called transfer RNA is this molecule. is going to go, so imagine this is all happening in a cell, right? This is this is your cell. Now, when you eat food, let's say you had a high protein, chicken Caesar wrap. So you've got a lot of protein, and the protein, it ends up sitting in your cytoplasm as a pool of amino acids, because that's what protein is made out of, right? Amino acids. So all your cells have a little pool of amino acids swimming around, okay? What tRNA molecules do is they're responsible for transferring amino acids from this pool to the mRNA strand, into the ribosome. Okay, now a few specific very important points about tRNA. It's made out of RNA. That's the first thing, right? So all of this that you see here, this is all RNA, single-stranded molecule, right? And at the very end, right here at the very bottom, we have what's called an anticodon. So all tRNA molecules have an anticodon. What an anticodon really is, it's just a codon. The reason it's called an anticodon is because it will always bind to its complementary codon. And to show that it's complementary, we use the word anti. So you can see here, if the mRNA sequence has a GAG, well, what's complementary to G, to C? What's complementary to A? It should be a T, but since it's RNA, it's a U, and then again G, so it's a C, right? So every single tRNA molecule has a different set of 3 letters, or a unique anticodon. So all of them have a unique anticodon. The next thing I want you to really understand is that the anticodon determines the kind of amino acid that is joined to the end of the tRNA molecule. Right? So tRNA again has an anticodon and it has an amino acid. Okay? So every tRNA molecule has a unique anticodon, and every anticodon has a specific amino acid, aka in the case of CUC, it could be the amino acid arginine is always going to be present. So in summary, what I've told you is that the mRNA sequence, A, always codes for a complementary tRNA sequence, anticodon sequence, B. And the tRNA anticodon sequence always has a specific amino acid sequence, C. So in summary, A controls B, B controls C. Simple maths, what conclusion can you all make? If A equals B, B equals to C, A equals to C. Right? The reason I wanted to highlight this was to show you, can you see how the mRNA sequence is directly determining the amino acid sequence of the protein, right? Whatever the mRNA codon is determining the tRNA anticodon, which is hence determining the specific amino acid that's sitting at the back of the tRNA molecule. Okay? Any questions so far? Okay, coming back to this image here. Again, we're just stressing the same point, but just showing you how it grows and it links, right? So again, the ribosome reads an mRNA codon sequence. It attaches a complementary tRNA that has a complementary anticodon, and that will have a specific amino acid on its back. What it will then do is, do you guys see the next position here? It will do the same thing. It'll read the next codon. It will attach another tRNA molecule with a complementary anticodon. And this is where the beauty happens. This is why you need to watch videos to really understand this. What then happens is the bond between the first tRNA and the amino acid will break off. So forget about this. Just imagine there was just one amino acid to keep it very simple. The bond here will break off, and a peptide bond will form between amino acid 1 and amino acid 2. And once that happens, everyone, the ribosome will then move one position to the right. And it will keep doing the same thing, and you can start to appreciate very slowly that an entirely new amino acid sequence is slowly going to be forming. And that, in essence, is translation. Now, this is a bit of extension that I'm going to be adding, okay? There is what's called a start codon, and there is what's called a stop codon in mRNA, okay? There are very specific codons that are start codons, very specific codons that are stop codons. You can search it up. In the case of start codons, it is always going to be an AUG, right? Every mRNA sequence, the first codon is always going to be AUG, okay? And what happens is AUG always codes for the amino acid methionine. Quite interesting. All proteins start with the amino acid methionine. That's because of the start codon that is AUG. Next thing, there are a couple of different variations of a stop codon. I think it's like UAA, UAG, and UGA, but double check that. But there are 3 different codons that code for what's called a stop codon. When a ribosome gets to a stop codon, Prithvi, do you know what it does? What does a ribosome do when it reaches a stop codon?
It doesn't attach like any more amino acids to it, so just the chain stops.
Good, but it attaches something. It attaches a protein. Does anyone know what protein it attaches to it? Again, this is a bit of extension, but in trials is very important because they give you a model of translation. If this isn't there, you can say this was a limitation. Okay, it makes a protein by the name of release factor. Release factor. And you can see that here, right? When you reach a stop codon, what happens? The tRNA does not bind. It's not nothing binds, but a protein called release factor binds. And what release factor will then do is it will go and it will snip the connection between the polypeptide and that tRNA. And that polypeptide will float away, and it will perform its role, or it will attach to another polypeptide to make a protein. Very good. Good. Okay. Now, final point to ask you all, what is the difference between a polypeptide and a protein? Britannia? Actually, I'll pass on to Atul. Atul, what is the difference between a protein and a polypeptide?
A protein constitutes many polypeptides.
One or more. There are some proteins that are so simple, they are just one chain of amino acids, right? There are also proteins that are so complex, they're made out of hundreds of individual chains of amino acids. Polypeptide is just a chain of amino acid, okay? And a protein is just equal to one or more polypeptides bound together. That's what a protein is. Good. All right. Any questions at all about polypeptide synthesis? After the break, I'm going to ask a couple of you guys at random to tell me an exam answer for explain polypeptide synthesis. Okay? Good. All right. Take your break. We will return at about 3:12, 3:17. Okay? So go get some water. Please move around. Look around as well, otherwise you'll fall asleep just looking at a screen, right? Yes, Atul,
Can
question?
you activate my EDX account?
Yes, I'll do that. I'll do that now. Okay. Thanks. Does anyone else log into EDX, the edX Learning website, and has an account? This is how you access your answers for the trial questions that I've given you. If not, in the break, how about all of you log into the edxlearning.online website? Once you do that, I need to approve your access for the biology resources, and then I can show you exactly where to go to access it. So can you all just quickly log in via your Gmail accounts? Just any account that I can see your name so I can give you access. Okay? So do that now, and I'll give you all of you access shortly. All right, see you guys at 3:17. Okay, when we're all back, can you guys hit the raise hand icon? Thank you. Great. All right. Sorry, guys. I might need to keep my camera off just for this week because my nose has started to bleed again. So I'm going to just keep it off for this week. But I apologize because I know that the engagement does stuff when you can't even see me. I can do anything about it. If it stops, I will turn the camera on again. Thank you for understanding. Okay. All right. If we're all back, did you guys all also log into the website and give yourself access? You hit the raise hand icon if you logged into the edX Learning website and you put your Gmail details in. Good. Let me show you how to actually access the resources too. Just remind me at the end of the lesson as well. Yeah. Okay. All right. So forget about this 6 marker. I want you all to just focus as if it was a 3-mark question, and it was explain the processes required to produce a polypeptide from DNA. I'm going to ask you guys at random to give me an exam answer, right? I don't want you to try writing up. You can, in this meantime, quickly think about how you're going to word it. But it's all about falling into the deep end, giving it a go. And if you can answer it verbally in an exam manner, then you can easily write it to get full marks in an exam as well. So, Ruthanya, whenever you're ready, you want to give us a little go. How about you give us a go with transcription? How would you explain transcription, Ruthanya?
It's the process of DNA making mRNA.
Remember, the question is explain. Is that all you're going to say? Okay, that's all right. Let's go to Matthew. Matthew, how do you know when you've actually answered an explained question?
I don't know. Cause and effect? I don't know.
That's what explained is. But Alyssa, how do you know when you look at your answer, Alyssa? Let's say you finished your exam, you're looking at your answer to an explained question. How do you know you've actually explained a concept? What words are you looking for?
Would it be like
That's all right.
a lot of like
Edward?
keywords?
Yeah, potentially. Edward, how do you know you've explained a concept?
Yeah, with like the relating cause and effect thing. So like as a result of and
Good. That's what I want to hear. See, there is a huge difference, guys, between the student that knows a lot of information and the student that does the best on exams. I want you guys to be balanced and good at both, especially the latter. Okay? So to do the latter, that's exam technique. To understand whether you've explained something, you need to know, you need to have questions or points in your answer, such as thus, due to, as a result. If you do not see that in your answer, or even because, if you don't see that in your answer, you haven't explained it. I don't care how many keywords you've used or whether you understand the question verb, you haven't answered the question. Right? So, very key important point. You have the HSC NASA verbs in front of you, which all of you should have printed, put on your wall by now. Now you need to start working on how you actually implemented it in an exam answer. So, Ruthania, you did good, but you didn't identify. I need much more detail. So I'm going to go to Pretty. Do you want to give it a go? Transcription. Can you explain it for us?
Transcription is the process where DNA helicase unwinds the DNA, and then an enzyme, RNA polymerase, builds a complementary strand. which is in the form of mRNA, and the mRNA exits the nucleus, the cytoplasm.
Good, good. I'd say that's good. I think the only thing you can do is maybe make it even more concise. Because if it's 3 marks, the way I'd break it down is 1 mark for transcription, 2 marks for translation. Because there's a bit more detail with translation, right? So one thing I'd also do is probably take out helicase. That's because if you look at the scientific papers, all of them mainly reinforce RNA polymerase. I've actually never seen helicase discussed in transcription itself. So I'm a bit wary about using that enzyme, right? So the key thing is I wanted to hear was RNA polymerase, right? And complementary strand. Okay, and possibly you can also include leaving via a nuclear pore. So something along the lines. Now, just to note, you don't need to have thus and due to in every single sentence line, as long as you have it in your overall answer. So in transcription, I don't really need to have it. But all I'm going to say is transcription involves the processing of a DNA template strand into a complementary mRNA sequence through the use of RNA polymerase. That's one line, but it has all the key words that we need to mention, right? Good. What about translation? Let's go to Anne. You want to give us an explain for translation? 2 marks.
I would probably say translation is the process by which mRNA that leaves through the nuclear pores connect to a ribosome, and the codons match up with anticodons of the tRNA to join to join amino acids that form a polypeptide, resulting in a protein.
Good.
Something like that.
Very good. I like how you put resulting in, just as you're explaining. What's the one word, additional word she could have included? She said matchup. What would be a better word to use?
Would it be complementary?
Exactly, right? Okay. So I think you're all starting to understand the word complementary is so important. Okay, very, very important. So I want to also hear that. But otherwise, very good job, man. Right? So as long as you're using those key buzzwords, and as long as you have due to, thus, as a result of, you're truly doing explain to a question. Good, good. Any questions at all about transcription and translation, everyone?
I've just got a question.
Yep.
So you know how the mRNA, do you say joins to a ribosome? What do you say, connects or?
Yeah, you can say the mRNA attaches to a ribosome.
Okay.
Or joins to a ribosome. That does not matter. Yeah.
All right. Thank you.
Or if you want to be fancy, you can say the mRNA positions itself within the ribosomal subunits, where each individual codon is bound to a complementary tRNA anticodon, which carries a specific amino acid sequence. Right. So just to give us, I think I have a sample answer. Right. I've literally given you all a sample answer for translation, just so you understand generally the detail, the buzzwords you need to be using. Right. And you can see here. Keywords I want to hear is mRNA, tRNA, ribosomes, codons, amino acid, polypeptide. Okay. And the word complementary as well. Good. Okay. If you have no further questions, let's move on to the next stop point. Okay, assessing the function and importance of polypeptide synthesis. Now, we've already gone through this. We've spoken about the structural and functional roles of proteins. Now, just a little bit more detail here. When you see an assess question, what does it mean? What do you have to do from an exam point of view? So I'm going to pass this on to Sarah, going to you. When you're assessing any concept in biology, what are you truly doing?
So basically, we're making a judgment, then adding the definition and the pros and cons.
Very nice, right? So the definition, I always say, include key definitions when it's 4 or more marks. in an exam question. Any big key buzzwords, underline it, include their definition. That might get you a mark, right? The judgment, pros and cons is perfect. That's exactly what assess is, right? So don't be the student that just thinks assess is provide judgment. You need to provide it against criteria, which are the positives and negatives. So I'm sure you all know all the pros of polypeptide synthesis. Can you guys state any, is there any way for us to state a con for the importance of polypeptide synthesis? How would you word that? You're not going to be saying a disadvantage of the importance of polypeptide synthesis is X, Y, Z. That doesn't make sense. But how would you word the con per se, where you're providing a statement against the absolute importance of polypeptide synthesis? Okay, let me help you all out here, right? Genes produce polypeptides. And we said polypeptides are important because they serve either a one structural role or they serve number two a functional role. I'm going to very quickly just go across and ask you guys some structural functional roles. And what's one structural role you're going to say in your exam? Just list it.
For structural, they make bones.
Good. Bone, connective tissue, right? So you'd say collagen specifically. You want to state the specific protein, right? Atul, next. What's another example? I want you guys all to be ready with an example. So you can do it fast. Good. What would you state as the enzyme?
Catalase.
Nice. Britannia, next. Structural or functional?
Was it neurotransmitters?
Very good, right? That's a functional role. Neurotransmitters involved in brain signaling pathways. That'd be perfect, right? I didn't tell you all neurotransmitter, but the main one you've all released this whole lesson, coded for in your consciousness, your learning, is glutamate, and it's a protein. Don't worry about memorizing that. I did not know it until university, so yeah, don't worry about that. Wow, time has flown. Not even in university anymore. Okay, structural, functional, I think you guys have got that set. The last thing I want to talk about is, well, what do those things serve? They determine who we are physically, right? And so the words we use for physical characteristics, the cumulative physical characteristics that make you up, is your phenotype. Remember, phenotype is your physical characteristics. And the reason we care so much about polypeptides is because they serve a structural and functional role that contributes to phenotype, right? If the protein is wonky or destroyed or mutated, then the structural or functional role it serves is also impaired, and the phenotype is diseased. That's how disease occurs. But let me ask you all, what is also very, very important in determining your physical characteristics? I'll give you all a hint. Yes, Edward, what was it?
Environment.
Environment, very good, right? So I want you all to remember this. Genes and the environment determine your phenotype. Okay, genes produce polypeptides and they contribute to phenotype, but the environment also does. Key example case, you would not have reached your growth potential if you grew up in a third world country with nutritional impairment, right? You'd be a shorter stature, your bones wouldn't be the same height, and it's not because of your genes, it's because of your environmental intake, right? There's much more interesting cases, but we don't have time to talk about that just now. So the way you could structure this, I'm gonna ask one of you. Let's go to Sarah. Sarah, how would you word this as a con for the importance of polypeptide synthesis?
So I would start off by saying, however, polypeptide synthesis comes with its cons, and then talk about the genes and all of that.
Yeah, good. That'd be a bit fluffy. And the thing is, guys, when I say pros and cons, I don't want you to literally use the word pros and cons in your answer. Okay? Is it doesn't sound right to say a con of polypeptide synthesis. The way I'd structure this is I'd say, although polypeptide synthesis is critical to phenotype, environment also serves a crucial role in determining the physical characteristics for an organism. Right? So do you see how I just worded it in such a way that I said that I pretty much pointed to polypeptide synthesis not being the all-important point, but other factors also being very important. Okay, so don't need to say con per se, but yeah, that's how I'd word it. Does anyone have any questions for this dot point? Pretty much every dot point you get, try doing an exam answer. So guys, when you're writing your notes, I would highly urge you, please do not write every single bit of information that you see in the textbook, or even to the depth of my notes. My notes are written in such a way that it's the first time you've come across a concept, and I go through everything from the basics to the full detail, right? Your notes, if you're writing your own notes, should literally be an exam dot point answer to that dot point. Very succinct, maximum, I think 3-4 dot points and diagrams, if anything. That would benefit you so much more than just doing this all over again. Okay, good. All right. Okay, now just one small point. What I've done here is to suggest the importance of polypeptide synthesis. I've spoken about a disease where improper polypeptide synthesis leads to disorder and impairs physical characteristics. So the one I picked is thalassemia. Thalassemia is a genetic disorder where you have a gene mutation, and that leads to a improper polypeptide. What is the protein that carries oxygen in our blood and contributes to its red color? Britannia?
Hemoglobin.
Hemoglobin. So hemoglobin, we abbreviate it as HB, looks like this. It's actually got 4 polypeptides. So each swirl that I'm showing you is actually a chain of polypeptides. It's got 2 polypeptides, and there's what's called 2 alpha chains and 2 beta chains. So 4 polypeptides in total. There are 4 different genes that each individually contribute to 1 polypeptide that makes up hemoglobin. What happens in thalassemia, you can have what's called alpha or beta thalassemia. There is a gene mutation in which we make an improper polypeptide, aka a mutated globin sequence, right? This is a globin sequence. Globin sequence just meaning each chain or each individual polypeptide chain of hemoglobin. So you can make a mutated alpha or beta globin polypeptide. And as a result, guys, what happens is you have reduced oxygen carrying capacity in blood, reduced O2 carrying capacity in blood. Does anyone have thalassemia here? It's awfully common among the East Asian population and also common among the South Asian population. So does anyone know if you have a family history or a family member affected by this condition? I can promise you one of your friends has some degree of thalassemia. It's either not diagnosed yet, and it's mild, but I can guarantee you. Yes?
Is this like when they have like low iron?
Yeah, low iron. That's right.
Yeah.
It actually,
my friends have it.
yeah, actually, and like they actually end up having normal or high iron, but lay people often say, "Oh, I have low iron," when in reality they have thalassemia. So these people either they need no treatment at all, or they need some mild transfusions of blood itself. But we won't go to the detail at this point. But good. Low iron is iron deficiency. So anemia is actually just a symptom. So because of this, they end up getting shortness of breath, tiredness, fatigue, and just a weird, interesting point. People who have iron deficiency or anemia, they get this weird urge to eat non-nutritional objects. I forgot the exact name for that condition, but some of my friends who have iron deficiency like eating ice cubes, right? Just a propensity to eat ice cubes, which is very interesting, but it's one of the symptoms for anemia. But that is thalassemia for you. Good. Good. Right. So again, I'm just stressing this. Whenever you are talking about polypeptide synthesis and its importance, the importance of genes, you must stress this point that it's genes and the environment collectively determine phenotype. And so that's what this whole dot point is. So I'll let you all quickly read this. Assess how genes in the environment affect phenotypic expression. So by now, you all know what assess means. You all understand what genes are. What we're now going to talk about is environment and phenotype. So going to Matthew, can you just remind the class, Matthew, what is phenotype?
It's a physical characteristic.
Very good. Now, interestingly, even though I said that just earlier, phenotype is physical characteristics, it's beyond just physical characteristics. It's also biochemical characteristics of an organism. So what I mean by that is if I created a clone of Matthew that looks just like him, exactly the same, but this clone has some form of disease inside the body, like anemia. You cannot see anemia on a person. You can't look at a person and say, you're anemic. You can examine them as a doctor and look for little signs, etc. But it's no clear physical telltale that you have anemia, right? But since it's still a biochemical impairment, we still say it's affected the phenotype. So that's just to explain to you, phenotype is physical and biochemical traits. So can you all remember that? And highlight the biochemical too for me. Very good. Now, what we're going to go through, everyone, are specific cases where the environment also affects your final physical and biochemical characteristics. Good. Has anyone already studied this dot point? How genes in the environment affect phenotype? Okay. Has anyone finished Module 5 at school? Module 5 biology? Okay, good. Because I was going to say, if your school's finished Mod 5, you've gone way too fast and you haven't actually grasped some of the concepts. Because this is probably one of the longest modules of HSC Biology. All right, cool. We'll go through this. All right, now before I do, I want you guys to have a quick read. I'm going to quickly blow my nose, be right back. Have a quick read of this, guys. Phenylketonuria, and then we're going to talk about it in about 20 seconds. Okay, thanks for waiting, everyone. All right, so let's talk about phenylketonuria, right? Sounds very complex. I'm sure some of you may have never heard of it before. Has anyone heard of this condition before? Okay. This tends to be a very interesting condition because it really highlights how important environment is in your physical characteristics. So let's break down what this word means, right? Phenylketone, right? This refers to a specific organic molecule. Phenyl meaning it has what's called a benzene ring. If you study chemistry, you'll eventually come across the benzene ring. It's just carbon and hydrogen. Ketones are a type of molecule. They're similar to acids. They're essentially acids. What does urea mean? Does anyone know? Urine, right? So phenylketonuria, what it really means is you're peeing out phenylketones, right? This specific chemical molecule. We'll talk about why that is the case. Now, normally, everyone, what ends up happening is when you eat different amino acids. This is you. This is your digestive system. Beautiful schematic drawing. You have multiple different amino acids. One of the amino acids that you eat and break down goes by the name phenylalanine. Okay, this is one of the 20 different amino acids that you have. Normally what happens is you have this enzyme called phenylalanine hydroxylase. And what it does is it breaks down or it adds a OH group. Can you see the difference between the molecule on the left and on the right? It adds an OH group. OH is the hydroxyl group, right? So the phenylalanine hydroxylase enzyme goes and slightly modifies and breaks down or changes the phenylalanine into a stable molecule tyrosine that's safe for us. What ends up happening for this phenylketonuria patients is they have a gene mutation where the gene that makes the enzyme phenylalanine hydroxylase is mutated. As a result of that, they don't make the enzyme phenylalanine hydroxylase. I want you guys to now tell me what would happen to the levels of phenylalanine and what would happen to the levels of tyrosine in this patient's blood and in this patient's urine. Hit the raise hand icon once you've got an answer. What would happen to the levels of tyrosine? What would happen to the levels of phenylalanine? Not too hard of a question. And what I'm literally asking you was one of the exam choice, I think it was the exam choice 2020 trial questions. A lot of students did not get this right, and I'll tell you why they didn't very shortly. It's not too hard, so don't think too hard about this. Good, Alyssa's got an answer. So this exact disease was asked in your HSC Biology trial paper. So just showing you, even though it sounds so weird and random, it is high yield. Okay, Alyssa, take it away. What would you expect to happen to tyrosine levels and phenylalanine levels in a patient's blood?
The phenylalanine levels would be really high and the tyrosine levels would be really low.
Yeah, exactly, right? So what students did in this trial paper was all of them were able to explain that tyrosine levels would be low, but none of them explained the high phenylalanine, which I know now all of you reading this would be like, oh, it's very simple, right? This line doesn't go across, so this is not made, and this increases. But I think in the stress of exams, some people forget to state some of the obvious key points. So very important that you remember and identify this, okay? Do you need to memorize this enzyme? Do you need to memorize all the different chemicals? The answer is no. I think the main thing you need to just remember is the buildup of phenylalanine, okay? And that would help if you remember the name of the disease, which is phenylketonuria. Phenylalanine is a phenylketone, right? So now it should make sense that if there's high levels of phenylalanine in the blood, there's going to be high levels of phenylalanine excreted in the urine, which is a phenylketone, hence the name of this disease, phenylketonuria, meaning peeing phenylketones. Now, you might be asking, well, so what? Who cares if a patient has high phenylalanine in their blood? The impact of this, everyone, is if your phenylalanine levels are too high, it travels to your brain and it causes seizures, mental retardation, and brain damage. Central nervous system, I'll draw the brain so you really understand that. My beautiful artistic diagram of the brain. Right. Okay, hopefully you can all kind of appreciate there's the little folds, the gyri of the brain. So you'll get mental retardation, seizures, epilepsy. Epilepsy is just recurrent seizures, so I'm not going to write that for us. Seizures. You'll also, very interestingly, these patients all get what's called like a musty body odor. It smells like old antique homes, right? Which is really weird, but that's the smell that all that high phenylalanine gives off. And question?
Yeah, I was just a bit confused. How is there like an increase in phenylalanine if it converts the amino acid phenylalanine into tyrosine?
The issue, remember, normally if you had, food that contains phenylalanine, your enzyme phenylalanine hydroxylase works, so it actually converts it into tyrosine, right?
Oh, okay.
These patients have a gene mutation where they don't have that enzyme, so all that phenylalanine builds up instead of converting to tyrosine.
Right. Okay, so they don't have the enzyme that converts it.
Yes, exactly. They do not have the enzyme phenylalanine hydroxylase. That's right. Okay, so these patients will get mental retardation, seizures, brain damage, intellectual disability, etc., etc. However, can anyone think of one way to stop this disease happening? Anyone? Don't.
If you don't eat the stuff that has.
Perfect. Exactly. Now, does anyone have Coke at home? A Coke can? Very weird question, but if anyone has a Coke can, go get your Coke can. The next time you drink a Coke, look at the Coke can and it will say contains phenylalanine. And now you will know why Coca-Cola has to put on every single can. Because if these patients drink this phenylalanine, they will get mental retardation, seizures, brain damage. But if they don't eat phenylalanine, aka they go to the doctor, they get a specific diet avoiding phenylalanine, they will live a perfectly normal, healthy life. That's how crazy this condition is, where environment is literally determining phenotype. So, of course, there is a gene that's mutated that's caused the disease, but it's the environment that is dictating whether you'll be normal or whether you will suffer a life of brain damage, mental retardation, seizures, etc. Interestingly, this disease is very common in Caucasian babies. Blonde-haired, blue-eyed babies are the most common demographic to get this genetic disorder. Any questions, anyone? I believe it's autosomal recessive. For those of you who've never heard of that before, don't worry, but it's passed down family trees. Good. Any other questions at all? I want this, guys, to be your main example of genes and the environment affecting phenotype. If you remember this one, you'll be set. If you use an example such as dietary intake and height, you're not going to get any marks because it's a very vague, almost could be thought up on the spot example. Okay? So have a very specific example of how genes and the environment affect phenotype. Any questions? Good. You can read up about the hydrangea plants. It's just pretty cool. They grow different colors in different soil. So soil pH is the environmental factor and color is a phenotype. I'm not going to go through that because you can read and learn it yourself. I'm here to teach you the hard stuff and how to think about biology. Pretty question?
Sorry, just making sure. How do you know if the person has a disease? Is it just like the symptoms of the brain, or?
Very good question. Very good question. First of all, do not say sorry asking questions because questions are the most important thing to learning. point for your question. Those of you who are born in Sydney, New South Wales, you would have had your heel pricked 3 times to obtain 3 drops of blood. And then what we do is we run that blood through genetic testing, which you'll learn very shortly. And that genetic testing will be able to pick up about 33 different genetic diseases, including phenylketonuria. And so you can imagine, Kriti, if a baby was born and we didn't know it had this and we gave it phenylalanine, it would get brain damage and it would be irreversible and it would be too late to help and save a life. So by doing this, this is called screening, where you're looking at the general population, you're looking for disease before it even comes on. That's what screening is. You can pick up these cases and treat them before they even get ill. The answer is genetic testing at birth is how we diagnose this condition. Good. This is called the New South Wales Newborn Screening Program. And a bit of a hint, this is going to be one of your examples for the end of this module when we talk about genetic testing. So it all links quite well. Good. Any other questions at all? Okay, let's go through. We're going to skip all this. We're going to skip epigenetics. All this, guys, is very extension. I didn't learn this until university. So read it in your spare time, but not too important. Okay, this stuff is highly important. So, Matthew, can I just get you to read the dot point?
Investigate the structure and function of proteins in living things.
Good. So I'm sure, Matthew, you know the function of proteins. You broke it down into structural functions and functional functions, right? Enzymes, neurotransmitters versus collagen and bone, actin and myosin proteins in muscle tissue, etc. Now what we're going to be talking about are the levels of protein structure, right? Now, when we talk about levels of protein structure, there are 4 levels, and I'm going to show you how they test you on this. So there is one very important point you should all remember at the end of today's lesson, but we'll quickly talk about it now. When we talk about levels of protein structure, there's primary, secondary, tertiary, and quaternary. And it goes to increasing complexity. You can imagine quaternary is definitely more complex than a primary structure. Primary structure is simply the amino acid sequence on the polypeptide chain, right? And we are talking about one polypeptide chain. Okay, that is primary protein structure. Okay, so you can see that right here. Primary protein structure is just whatever sequence this polypeptide chain has. Now, what you can all appreciate happens is after there is a primary structure for a protein, it starts to form a unique 3-dimensional shape. You can see what's happening here is it can either, the polypeptide sequence can either move up, down, kind of like a corrugated roof you see in those old houses or the sheds you might see at school. Some of the roofs might look like this, right? Where it goes up, down, up, down. What's essentially happening there is parts of the polypeptide sequence are facing up, parts of them are angling down, and then another section here will move up, another section will move down, section will move up, section will move down. Right? And the reason this happens, guys, is because of hydrogen bonding in the sequence. So you can see there could be, for example, hydrogen bonds, weak hydrogen bonds between that and that amino acid that will help form this roof, this sheet-like shape. This is also known as a beta sheet, so you'll see that. Another arrangement it can form is kind of like DNA, but instead of a double helix, a single helix, where it arranges itself in this form. And this is called an alpha helix. So when you talk about secondary protein structure, key thing is due to hydrogen bonding within that single polypeptide chain, and it can form a beta sheet or an alpha helix. Good. What can then happen, guys, is this alpha sheet and beta helix can further fold in a very 3-dimensional nature. Now have a look at this, right? When you zoom in here, you can see, what is this? Going up, down, like this. Rithania, what do you call that? Is that a alpha helix or a beta sheet?
Beta sheet.
Very good. And then you can see here, it switches. Remember, this is still one polypeptide chain. It switches to become helical. It's going in a helical arrangement. But can you see that the alpha helix and beta sheet have further folded up into the page, out of the page, into a complex three-dimensional shape? That is called tertiary folding. It's when you start folding the sheet and the helix into a 3D shape. This is still one polypeptide chain. This is called tertiary protein, and the buzzword I want you to state is 3D folding. Okay? Now, here it forms some stronger bonds. There are specific bonds like disulfide bridges that help form these bonds. There's also hydrogen bonding that also occurs to help form this. You don't need to memorize this stuff, but just to explain to you why it's forming this shape. Finally, quaternary protein. This is the most important thing. To have a quaternary protein structure, you need more than one polypeptide chain. More than one polypeptide chain. The second you've got more than one polypeptide chain, it is immediately quaternary. So hemoglobin, what level would it be? Primary, secondary, tertiary, or quaternary? This was a MCQ question with one of the trial papers. What they did was they told you the structure of hemoglobin, which I'll remind you again because we went through this. What level of structure is hemoglobin?
Quaternary.
Very good, right? That is the most important one because that's how they'll test you. Okay, very good. All right, guys, that ends it for today. Do we have any other questions? So right now we're on page 112. We're making good progress. We just need to now talk about inheritance and genetic variation. So we'll probably finish Mod 5 in about by the end of the term. Good. All right. We have no further questions. I will see you all. Yes, question.
Can you activate my account?
Yeah. I'll get onto that. Until I have a class right after this as well. But once that's done, I'll activate all of your accounts. Okay. Good. All right, guys. I will see you next week. All your accounts will be activated tonight as soon as I finish my classes. But have a good week, and please check the Teams channel. By tonight, I'm going to tell you the date for our makeup class this coming week based on your availabilities. Okay, so please check it tomorrow on Teams so you attend on time. All right, have a good week. See you guys.
Thank you.
Bye. Bye, everyone.
Sir?
Yes.
Sir, I just wanted to recheck, is the website edxlearning.online the one that we need to sign into?
Yes, that's right.
All right, thank you so much.
No worries. Have a good week.
You too.
Bye.